Welcome back to the third day of our National Faculty Development Program on Emerging Trends in Intellectual Property Rights, organized by Vimla College Autonomous Thrissur in collaboration with the Inter-University Center for IPR Studies, Cochin University of Science and Technology, Ratnavel Subramanian College of Arts and Science, Coimbatore, and PSGR Krishnamal College for Women, Coimbatore. Let's begin today evening with a silent prayer. Thank you, dear friends. I now call upon Dr. Shija T. Taragan, Associate Professor and Head, Department of Botany, College, to welcome us all and introduce the resource person for the day. Thank you, ma'am.
Thank you, Maliga. Am I audible?
You are, ma'am. Quite
Yes.
audible.
Okay, thank you. Good evening, everyone. It is with a great pleasure that I welcome you all to this faculty development program about intellectual property rights. We are truly honored to have a distinguished resource person, Dr. Asha R., who will undoubtedly enrich our understanding of this vital field. Today, she will be handling the topic patenting of biotechnological inventions. Let me introduce her. Dr. Asha has been serving as a research officer at the IPR Facilitation Cell of the Inter-University Center for IPR Studies at CUSAT since July 2018. Her academic journey began with bachelor's and master's degree in chemistry from Kerala University, followed by doctoral degree from the prestigious Rajiv Gandhi Center for Biotechnology in Trivandrum, Kerala. During her doctoral studies, she was a recipient of the CSIR Fellowship. Her contributions to the field of science are exemplified by her numerous research publications in international peer-reviewed journals with a focus on antimicrobial peptides. Dr. Asha's dedication to academic excellence extends beyond her research achievements. She has been an active participant in various national and international conferences where she has presented her work and contributed to enriching the global scientific discourse. Her achievements also include being a qualified DST Women Scientist C and a recipient of the DBT Travel Grant, highlighting her commitment to advancing her field through continuous learning and collaboration. One of Dr. Asha's notable accomplishments is her role as a registered Indian patent agent. She is deeply involved in facilitating various aspects of patenting, from searching to filing and drafting. Her expertise in this area has led her to become a successful resource person, contributing her insights to numerous IPR webinars and workshops. Moreover, her association with various national bodies focused on IP-related matters further emphasize her dedication to the advancement of intellectual property rights in our country. We are truly privileged to have Dr. Asha with us as the resource person for this faculty development program. Her wealth of knowledge and experience will undoubtedly provide us with invaluable insights into the world of intellectual property rights. On behalf of the entire organizing committee and all the participants, it is my pleasure to extend a heartfelt welcome to Dr. Asha R. to the Faculty Development Program. I also extend a sincere welcome to all the participants who have chosen to be the part of this enriching program. Let us seize this opportunity to expand our horizons, deepen our understanding, and collectively contribute to the growth and the dissemination of the knowledge in the field of intellectual property rights. Once again, a warm welcome, Asha ma'am, to the session. Thank you.
Thank you, ma'am. Thank you for your wonderful introduction. So, I'm really thankful to all the organizers of this FDP. So, this is the third day, I believe. So, you must be here. You must have gained knowledge about the introduction to IP. So, today I'll be dealing with a more specific topic, which is the patentability of biotechnology inventions. So, let me share my screen. Just a minute. Is it visible?
Yes, ma'am.
Yeah. So today my agenda will be like, I'll be dealing with a brief about patents. Then I'll be dealing with the biotechnology, what is biotechnology and how it is classified, what are the areas of biotechnology. And after that, I'll move on to the patentability of biotechnology invention, why it is so relevant these days. And then the claims in the biotechnology invention, how the biotechnology inventions are actually structured. Then I'll move on to the patentability criteria of biotechnology inventions. And after that, I'll be briefing about my center also. So that is my agenda today. So welcome all. So how many of you are from science background? Can you raise your hands? Yeah, good. So because this is more of a technical topic. Okay, so let's move on to our topic today. So it's patentability of biotechnological inventions. So what is a patent? So you must heard about it from the first session, I guess. It was on the overview of the intellectual property rights. So patent is actually a right which is given for an invention. So if you disclose your invention to the patent office, they will give you a right, and it is for 20 years. So that is a limited monopoly. For 20 years, you'll get a limited monopoly. So that is patent. Clear? So it's a statutory right for an invention granted for a limited period of to the patentee, that is the inventor or the applicant, in exchange of full disclosure of his or her invention. And that particular right will give you some particular rights. What are the rights? You can exclude others. Okay, you can exclude others from making or using your invention, or importing or selling your patented product or process for producing that product for purpose without his consent. That means without your consent, anyone cannot use your invention. Okay, that is the right you are getting from this particular patent. So it is a monopoly, but it is a limited monopoly for only for 20 years. And there are some exceptions also, in these 20 years, that is what you cannot say it as a permanent monopoly. It's like the government, it can intervene in between. If there is a situation, government can intervene and government can use your invention. That is, there are some situations are like that. The compulsory licensing situation and this BOLA provisions are also there. But it is a limited monopoly. Okay, it's a monopoly. You have to understand that it's a monopoly. Even if it is limited also, it is a monopoly. And how will you get a patent? So if you have invented something, you can get a patent. How you will get a patent? So these are the criteria. These are the standards of patentability. So it should not fall under the section 3 and 4 of Patent Act. Okay. And the other three criteria are novelty, inventive step, and industrial applicability. So novelty means it should be new. Industrial applicability means it is capable of using in an industry, or it should have some use. And inventive step, that is the most important criteria. So what is that actually? So according to the Act, what is inventive step means? If your invention if your invention has to be inventive, there should be some technical advancement with respect to the existing knowledge. Okay. So that is inventive step. Otherwise, there should be some economic significance or both. That means both this economic significance and technical advancement. And that should not be obvious to a person skilled in the art. That means if you're making an invention and it is having, just say if you are actually have some invention with your hand, and it is already it's just imagine that it is an improvement of an already existing knowledge. So then you have to prove that there is some technical advancement for your invention with respect to the existing invention. You just say, imagine that there is a cancer drug and I am coming in with a new cancer drug, and I'm saying that this particular cancer drug has got some additional effect. That is, this has some, targeted action. So if you consume this kind of tablet, there is no side effects. So I'm coming in with such an invention. So there is a technical advancement compared to the other existing cancer drug. That is having this chemo drug having this side effects. A lot of side effects are there. So my drug is, I am coming in with this new drug and it is not having any side effects. That is my technical advancement. So that kind of an advancement should be there in your invention. Then it is actually, we can say that it is inventive. Clear? Okay. So I'm not expecting, I think I won't expect answer from you since it's an online session. So now I will move on to the biotechnology inventions. So what is biotechnology? What is biotechnology? It's actually the manipulation of biology or the technology associated with biology, right? So we are actually manipulating the living systems, living organisms, or the normal biological processes for making some advancements. Okay. So it is actually dealing with the living systems. So that is why biotechnological inventions are so relevant nowadays. So if we are giving some monopoly to this kind of inventions, what will happen? We're actually giving monopoly to life, right? So that will not happen. So that is why this topic is very relevant nowadays. So why biotechnological inventions are relevant? So because there is one case related to this particular area, that is the Myriad Genetics case. So in that particular case, what happened was, the BRCA1 gene, BRCA1 and BRCA2 genes. These, if there is a mutation in these two genes, you will have a chance of getting getting breast cancer or ovarian cancer is high if there is a mutation. So what happened was, this Myriad is only actually it was Myriad Genetics is a startup which is based in Utah University, and they've actually they have discovered these two genes, and they tried to patent these two genes. And what happened was, they were only a startup, and after 10 or 15 years, what happened was they became a multi-billion company. So only because of this particular patent. So they have patented these two genes, and they have developed actually a diagnostic method to detect the possibility of getting cancer or breast cancer or ovarian cancer in women. So that is actually that is a landmark case in this biotechnological invention. So in that case, they actually patented two genes. It is actually a naturally occurring thing. It is a living thing. So they have actually patented a living thing. Is it possible? We cannot patent living things, right? But they have actually patented this thing, and they have there are a lot of there was a case with this Association of Molecular Genetics versus Myriad. So then they have actually opposed this patent, and finally what happened was, they've given patent only for cDNA. That is a complementary DNA, and they are saying that this complementary DNA is patentable, but the naturally occurring DNA is not patentable. So it's also a debatable topic. We'll discuss it later. Okay. So biotechnology is the manipulation of living organisms and systems and processes for benefit of the society, the environment, and the industry. So mainly there are four types. We have actually classified this biotechnology into four classes. That is red biotechnology, green biotechnology, white biotechnology, and blue biotechnology. So what is all these things? It's like simply if it is application of this biotechnological processing in the area of medicines. If it is an area of medicine, it is called red biotechnology. So nowadays it's very relevant because all these cancer medicines are biologics. So red biotechnology is very relevant nowadays, and it's very costly also. All these biologic medicines are very costly. There is one drug called Humira. It is actually used for I think rheumatoid fever, actually, rheumatic arthritis. So it is actually for that particular disease, and it is having more than 138 patents for single invention, actually, single drug. So they have actually got 138 patents, and they have a monopoly till I think around 2035 or 40. So that is the power of patent. They've actually they actually protected that particular drug through 138 patents. Okay, so biologics are very important nowadays. That is, so that deals with the red biotechnology. And if the biotechnological application is in the area of agriculture, it is called as green biotechnology. And if it is in the marine systems, it is called blue biotechnology. And if it is in the industrial process, like food industry or plastic industry or something like that, then it is called white biotechnology. Okay, this is very brief about biotechnology. So biotechnological inventions are like this, it can be products and process of gene engineering technologies. It can be methods of producing organisms, or methods of isolation of organism from culture medium, or methods of mutation of cultures, mutants, transformants, plasmids, etc., or processes for making monoclonal antibodies, or cell lines for making monoclonal antibodies. So these are actually the upcoming areas of biologic drugs, these monoclonal antibodies. You can, the famous drug for cancer, the chemotherapy drug, trastuzumab, is also a monoclonal antibody. So it's actually used for chemotherapy. So all these are antibodies, monoclonal antibodies. So, so biotechnology inventions can be of these categories, like products and process of gene engineering, or methods of producing new organisms, like genetic engineering, or genetically engineered organisms, like genetically engineered bacteria, and methods of isolation of microorganisms from culture medium. So here, that is also a gray area, methods of isolation of the culture from culture medium, because it's simply isolating a bacterium from a medium is actually, it's a debatable area. And methods of mutation. So how you are mutating a particular gene, and process of making this MABs, that is the monoclonal antibodies. So these are actually categories of biotechnological inventions. These can be biotechnological inventions. So to begin with, so we need to understand the history of this, the patentability. Okay. So when we introduce this patentability system in our system, our country, so TRIPS, the trade-related aspects of intellectual property rights, we have actually adopted our patent law from this TRIPS. Actually, we made it consistent with TRIPS. So they actually mentioning they are saying that, so we have to give patent, subject to the provisions of paragraph 2 and 3, patents shall be available for any inventions, whether products or process, in all fields of technology. So that is what TRIPS language is. You need to give patent to all fields of technology. You cannot discriminate technology areas. Okay, so you have to give patents to all fields of technology. So then there will be a problem, like, right? If you want to patent everything, if you have to patent everything, so what will happen? So everything will be under monopoly. So TRIPS also given some flexibility. So they are saying that members may exclude from patentability inventions the prevention within their territory. And so they have actually, they have given some freedom to exclude some categories of inventions from patentability. So that is actually the excluded patentable subject matter. So they're saying that necessary to protect the order public or morality, including to protect human, animal, or plant life or health, or to avoid serious prejudice to environment, provided such that the exclusion not merely because the exploitation problem by the law. So if an invention is actually prejudiced to the human health or animal health, you can exclude that thing from patentability. Also, you can exclude from patentability that what although the diagnostic, therapeutic, and surgical methods. So if you are patent the surgical methods or therapeutic methods, what will happen? The treatments will be very costly, right? Even now also it's very costly. But if you are included all these things, like the treatment methods, the diagnostic methods, so all will be very costly, right? So members have the freedom. So countries can decide what all things you have to include in the patentability criteria. And in US, these things are patentable. So they have included what diagnostic methods, method of treatment are everything under patentability. But in India, actually we are excluded from patentability. And they have said that you can exclude plants and animals other than microorganisms and essentially biological processes for the production of plants or animals other than non-biological microbiological process. So they've actually excluded these many things from patentability. So based on this, in India till 2002, we were not granting patents for living entities of natural or artificial origin, biological materials or other materials having replicating properties, substances derived from such materials, and any process for the production of living substances, entities including nucleic acid. So we were not giving patents to these kind of things till 2002. But in 2002, we have amended our law and we have included this biochemical, biotechnological, and microbiological processes were included within the scope of chemical processes for the grant of patent. So we have actually included the process, not the product. We included the process or all the microbiological process, biotechnological process into the category of chemical process. So thereby we have included some biotechnological invention in the patentability. Okay. So initially we were not giving patents to these areas, and we have the freedom to exclude all these things from patentability. Okay, and we have excluded that also. So in 2001, we actually signed this Budapest Treaty. So what actually this treaty mandate is that, so whenever you are actually planning to patent a process related to a biological substance or a biological material, so you need to deposit it into some depositories. There are some depositories which is actually recognized by this treaty. So it's IMTech Hyderabad, then MTCC. All these things are actually recognized as the depositories. So if we want to patent an invention which is related to a microbiological material, so you need to deposit that particular thing in this depositories. And also if you are using some natural things like biological resources from India, so you need to get the approval from the National Biodiversity Authority. Then only you'll get a patent. Okay, all these things came into picture. So if you want to assess the patentability of a biotechnological invention, so what are things we have to look into? So like every other invention, so I told you earlier that, so if you want to patent something, you need to satisfy 3 conditions. That what are they? Novelty, inventive step, and utility or industrial application. And it should not fall under this section 3 and 4. These are the criteria to get a patent. So the same criteria is applicable here in biotechnological inventions also. Okay. So, and what are the sections actually deals with the biotechnological invention? The exclusions, like in section 3 actually deals with the excluded subject matter. So what all excluded subject matter are relevant to the particular this patentability of biotechnological invention. So what all they? It's inventions contrary to the section 3B, section 3C, section 3D, 3E, 3H, 3I, 3J. I'll discuss everything in detail, okay? And this computer programs, 3P, then 10 clause 4, actually the sufficiency of disclosure. So we used to say that actually, the patentability criteria are actually 3, but it's you have to say it as actually 5. So the novelty, inventive step, utility, and then the section 3(4) and exclusion and the sufficiency requirement. So if you are not sufficiently describe your invention in the, description, it is not patentable. Okay. And unity of invention. So what is unity of invention? It's one patent for one invention. That is unity of invention. So all these criteria should be satisfied. Okay. So we'll move on to each and everything. And before that, we need to understand what are the claims actually in related to this. What? Is it visible? Yes. Okay. So what all the claims actually structured? You know, when you file a patent, how the claims are structured in a biotechnological invention. So it must be, it will be like the polynucleotides or gene sequences, or polypeptides or protein sequences, or vectors or plasmids. Either it can be gene construct or cassettes or gene libraries. So you can see all these words in biotechnological patents. Or host cells, or microorganisms and stem cells, transgenic cells, plants and animal tissue cultures. So all these things are actually used in biotechnological inventions. Okay. So when you assess the novelty of the novelty of biotechnological invention, so it is just like any other subject matter, any other type of inventions. So if it is patentable if it is new. So it should not be anticipated by a prior art. Okay. So that, what does it mean actually? So what is actually novelty? See, I'm having a pen. So this is my invention. Okay. This is my invention. I'm going to patent this particular invention. So I am having a different type of pen, which is actually there in the prior art. Okay. And just imagine that this is a ballpoint pen, and my prior art is actually a ink pen. Okay, but ink pen is already there, and I am coming up with this particular ballpoint pen. So whether it is novel or not. So when you assess novelty, what should you do? You have to list the features of this particular invention. So this is your invention. You have to list the features of your invention. So this is a ballpoint pen. This is having there is a ball at the tip of this pen, which will move, and which is having an ink, which is not like the ink pen. It's this ink is having more viscosity than the other ink, which is used in the other pen. So all these features are there, right? So you have to compare these features of this invention with the prior art. So the prior art is your normal ink pen. Okay. If you are getting all the features exactly similar in the prior art, just imagine that situation. So everything is same in the prior art. So then we can say that this is not novel. But in this situation, what is happening actually? This ink pen is not having all the features of this particular pen. Okay. So this particular thing is novel. So that is the novelty criteria, whether it is there or not. That is novelty. Okay. So you need to find out the prior arts, and you have to match the prior art. So in a single prior art, in the case of novelty, it should be in a single prior art. Okay. So in a single document, you have to match everything. So this particular pen, the ink pen is my prior art. I got the prior art as ink pen. So I have to match the features, and I have found that this is novel because the features of this pens are this particular pen is different from the ink pen. But there are some similarities also, right? So this maybe the shape of this pen may be the same, or the mechanism, like there is an ink, but there are some differences also. So that differences make this particular inventions novel. Okay. So in the case of this, different types of claims. Okay. A claim to product obtained by a process is anticipated by disclosure of only that product per se. Okay. If it is a product by process claim, so now it is not allowed in India, this product by process claims. Earlier it was there, the product by process claim. So claimed product obtained by a process. So we are actually claiming a product and which is obtained by a particular process. That is also you have mentioned in the claim. And is anticipated by disclosure of only that product per se. That means if there is a disclosure of a product, the same product, then also this invention is anticipated, regardless of the method of production. So you are actually claiming a product obtained by a process. Okay. And the prior art is the same product is there, but it is a different method of production. So that particular invention is anticipated because the same product is claiming. Is that clear? Okay. So when come to the sequence claim, so claim to polynucleotide sequence that was available as part of library before priority date lacks novelty, even if function has not been previously determined. So that means there is a DNA sequence or a nucleic acid sequence or a peptide sequence. Imagine that there is a sequence. Okay, you are claiming a new sequence. And you are claiming this particular sequence for a new function. So which is now we are claiming it as this particular sequence having a new function. But this particular sequence is already available in the library or in the database. It is already there. Okay, in the NCBI it is already there. And you are claiming a new function of this particular same sequence. That is also it actually affects novelty because the particular sequence is already known and you are actually only claiming a new use of this particular sequence. Okay, that is also affect novelty. So this is all these things are happening in the area of biotechnology. And composition claims. In case of composition claim, what is composition? There may be one or two substances, like if it is a tablet, and tablet must be having the active ingredient, the inactive ingredients, all those things are there. So all these things together called a composition. So a composition useful against diphtheria toxin, comprising antibodies together with acceptable preservatives and stabilizers. All these things are what? Excipients, the non-active ingredients, wherein antibodies are obtained from chicken egg yolk. Okay, they are claiming a composition of antibodies, which is actually a composition with preservatives and stabilizers. So antibodies we used to preserve it like that. Okay, all the vaccines and all those things that we used to preserve it like that with stabilizers and preservatives. So what is the prior art here? A composition of useful against diphtheria toxin, comprising antibodies obtained from chicken egg yolk along with additives. Okay, also discloses methods of making egg yolk antibodies. So here actually they are, claiming a chicken egg yolk along with other additives. So this is also a anticipating disclosure. Okay, this affects novelty of this particular invention. Is that clear? Okay. So now we are moving to the inventive step. So how you will assess the inventive step in biotechnological invention? As I mentioned earlier, like inventive step means there should be some technical advancement or having some economic significance and makes the invention obvious to a person, not obvious to a person skilled in the art. So what does actually means? So like I pointed earlier, like if there is a drug, it was an anti-cancer drug, and which is having a lot of side effects. I'm coming up with a new drug which is having a targeted action. So that is having a technical advancement. So I can prove that, right? So initial what all the initial drug is having this effect, like you'll lose your hair, you'll be having bleeding, lot of side effects will be there. So but this new product is having some technical advancement. That means it does not cause this kind of side effects, which is having a targeted action. So only kill the cancer cells. It's only go to that particular targeted site. That is a technical advancement here. Okay. So like that, in biotechnological inventions also, we need to evaluate that inventory step. Okay. So simply we are granting patents in biotechnology will lead to actually it is actually against the human life. So we need to examine this kind of invention very carefully. So suppose an isolated DNA sequence encoding a mature human IL-3 protein having a proline residue at position 8 of the mature polypeptide set protein processing bond proliferation inducing activity in human bone marrow proliferation assay. So this is an invention. Okay. It was mentioned that the difference with the prior art is that the claim compound is at position 8. There was a proline moiety, whereas in the prior art, there is a serine moiety. So instead of proline in the prior art, it's only serine. So what is the difference? It's only an amino acid difference, right? So does that make any difference? I'm not a biotechnologist, but I'll say that. A single amino acid change might have, some effect on the function of this protein, even if it is if it is in the critical regions of this protein. But if it is not in the critical region, there will not be any effect of this particular change. Instead of proline, there is serine. So when analyzed by the control, it was found that the single variation in the amino acid sequence does not normally change the activity and function of the protein unless its change is in the critical region of the protein. So, and the applicant could not prove any evidence that the protein coded by the claimed DNA was different from the prior art and in its chemical properties, and inventory step was not acknowledged. So, if you want to just take the situation of the same invention. So, if you wanted to patent this particular invention, you have to prove that the change in the position 8 with proline has made some significant effect on this function of this particular protein, and you have to show with some assay. Okay, so you have to show that with the serine molecule and with the proline molecule. So what is actually the change? So you have to prove that this much change is there. Then you can get a patent. Okay, so that is simply inventive step. Then industrial applicability. So industrial applicability, according to the act, they have actually mentioned it as capable of being made or used in an industry. So if there is an invention, it should be capable of being made or used in an industry. It should have some use. And according to section 64, they are saying that if it is not useful, it will be actually revocable. If you want to revoke a patent, you can prove that if there is no use for your invention, it is actually a ground for revocation. Okay, and specification should disclose the usefulness and industrial applicability of an invention in a distinct and credible manner. So when you file a patent, so you have to disclose the utility of your invention in the description itself. So it should be very clear. So what is the application of this particular invention? But you don't get a patent for this. You cannot claim the application. You are actually claiming the product, and the usefulness of the product should be there in the description. Okay. So just imagine this situation, that is a polypeptide in substantially isolated form whose sequence is shown in any one of CQIDs 1, 3 to 32, 36, 46, and 47, or whose sequence is encoded in a polynucleotide selectivity hybridizable with polynucleotide as shown in any of the sequence ID 1, 3, 32, 36, 30, 46, or 47. So they are actually, in this particular invention, they have actually shown many sequences, many polypeptide sequences. So it's usually like that. If it is a biotechnological invention, so what all things they will claim, they will claim the nucleic acid sequence, and they will claim the corresponding amino acid sequence also. They'll list all the sequences in the patent document. Okay, like that they will do that. that is a strategy of this companies. Okay. So in this particular invention, what happened was, upon examination, it is seen that the said claim covered an almost vast number of polypeptides for which no use was established. Okay, and the said claim therefore was not allowable on the ground that it lacked industrial applicability. They have claimed a lot of compounds, right? They have claimed a hell number of what polypeptides or nucleic acid sequences. So many things are there in the description, but they have not established the usefulness of this kind of sequences. They might have exemplified one or two sequences, like they might have given the application of one or two sequences, and all other sequences are actually the possibilities. They're actually disclosing everything, and only the sequences, not any usefulness of this particular sequence. And since it is not useful, it is not patentable. Okay, clear? So there should be a use also. So we have discussed the novelty, the inventive step, and utility. Okay, that is industrial applicability. So if an invention should be patentable, it should satisfy all the three conditions. That is novelty condition, and the inventive step, and the utility, the industrial applicability. Okay. So now we move on to the subject, the exclusions with respect to the biotech inventions. So Section 3B of Indian Patent Act actually deals with what? The inventions, actually, Section 3 and 4 actually say that some areas are not considered as inventions. So inventions contrary to morality or which cause serious prejudice to human, animal, or plant life, or health, or environment. Okay. So it is actually inventions which is contrary to morality or which cause serious prejudice to human, animal, or plant life, or plant health or animal health. It is actually not an invention according to Indian Patent Act. So this is very relevant in the case of biotech inventions. So process for cloning of human beings or animals. There are a lot of cloning is happening nowadays. So in China or something, I think in China only they have cloned human babies also. So not cloned, actually, they have actually engineered this human sperm, and they've actually, with the human engineered sperm, they have actually made a baby. So all these things are happening nowadays. So in India, we are actually prohibiting these things as non-patentable subject matters. So process of modifying the germline of human beings. Process of modifying the genetic identity of animals which are likely to cause them suffering without any substantial or medical or other benefit to man or animal, and also animals resulting from such process. So why this, gene editing technology? Now it is like CRISPR and CRISPR Gold, everything is there in the pipeline. So a lot of things are happening. So all these things are actually, it's useful in some extent, but it's actually there are a lot of ethical and moral issues, right? So cloning is actually prohibited from patentability, human beings or animals. But in some countries, they are actually, they have actually, admitted this cloning of animals. So they have made Dolly, the sheep. And in US, there is a case called OncoMouse case. So they have actually, made a mouse model for cancer, for cancer studies, and which is actually it's a living thing, right? And they have actually manipulated the gene of this particular mice. And if you are modifying this kind of mutations in this animals, you will get a modified mice which you can use as a study model. So that is actually an issue with patentability in US and I think in Europe, I guess. So, and they have actually given patent to this particular invention. But when they have whereas one more case was there, but in that case they haven't given patent. So there it's actually vary in case to case basis. So in India, we are actually excluded this from patentability. Use of human embryos for commercial exploitation. So there are a lot of there are a lot of rumors are there that human embryos are used in cosmetic industries like that. So if they are using such compositions in cosmetic industry, such things are not patentable. Okay. Then discovery of any living or non-living substances occurring in nature are not patentable. So living things are not patentable. So, or non-living things occurring in nature are also not patentable. Okay. So just for example, you are isolating a bacteria from a soil. Is that patentable? No. It's simply an isolation from from the nature. It's just a discovery, right? Discovery, you won't get patent for discovery. So aren't there conventions international against? There is a question. Aren't there conventions international against such living material patent? Yeah. So I already mentioned that. See, in TRIPS itself, they are actually giving the freedom. So they are saying that members may exclude from patentability, and they are not saying that you should exclude. So they have actually given freedom to the countries. Okay, so that is why we are actually giving this living things patents in different countries. The different countries have different positions. In Europe, they have this lot of ethical issues, but in US, they are not looking into the ethical issues. They'll give patent left and right. Even because they have given the freedom of excluding from patentability. So what all things exclude, they can decide. So they have the freedom, but they are not mandating it. So they are saying the TRIPS languages members may exclude, not should exclude or shall exclude. They are saying that may exclude from patentability. So discovery of any living or non-living substance occurring in nature is not patentable. So products such as microorganisms, nucleic acid sequences, proteins, enzymes, compounds which are directly isolated from nature are not patentable. So if you are isolating a product from nature, so just imagine that you are isolated a compound from nature, which is an anti-cancer compound, which is not patentable. So if you are actually making some modifications to that particular product, or you are claiming it as a composition, it is patentable. That is the thing. So then natural things also can be patentable. So if you are having a good attorney and a good patent agent, you will get a patent for anything. So that is actually happens in India. So a novel agent for promoting cardiac development activities, that agent having CQID number 1, wherein the agent is obtained from the perivitrilline fluid of horseshoe crab, Tachyples gigas. Okay, so this is a claim for an invention. So a novel agent promoting cardiac development activity, and it is obtained from the perivitrilline fluid of horseshoe crab. It is actually isolated from this particular organism. Okay. So it is not patentable under Section 3C of the Act because the claim attempts to claim an agent which is isolated from the perivitrilline fluids of embryos of horseshoe crab. That is a peptide which is non-living substance occurring in nature. So it is a non-living substance which is isolated from nature, and it is not patentable. Okay. So that is the thing. So it is a non-living substance which is occurring in the nature, and just you are isolated from it. It's not an invention, it's only a discovery. Clear? So now we will move on to the next section, that is the 3D. It is actually the most controversial section of Indian Patent Act. There is one more question. So can an Indian scientist apply for patency then in those countries which permit? Yeah, yes, sure, you can apply. No issues. So patents are territorial in nature. So if you cannot patent it here in India, you can patent in any other country if it is allowable in that country. In US, it is actually, they have given this patent for oncomouse and given this patent for this BRCA1 gene. So it's a lot of things are happening. So they are actually giving patents for MABs. So if you're not getting patent here, you can patent there, but you will get the right only in US. So it's territorial nature. That means if you have a patent in India, it is enforceable only in India. You can get a patent in US in that situation. And if you have a patent in US, it is only enforceable in US. You cannot come in India and say that I have a patent in this particular area, so you cannot use this particular animal. So that you cannot say because you don't have a right in India. You only have a right in US. That is the territorial nature of patent rights. I think I have answered your question. So, so mere discovery of a new form of a non-substance which does not result in enhancement of non-efficacy or mere discovery of any new property of new use of a non-substance. So what does it mean? It's actually, this is the most controversial section of Indian Patent Act. Why we have introduced this particular section? Because, in the area of pharmaceuticals, so what is actually happening? So all these medicines are, very highly priced. I can give you an example. There is this particular drug called Herceptin, which is used for this triple negative breast cancer. And it is actually initially when it is introduced during the patent term. So they have the price of around 1,13,000 per injection. Okay, just imagine this. It's 1,13,000 per injection, and a person should undergo 14 such injections to complete a course. So imagine the cost associated. So that is the problem with the pharmaceuticals. So if they have a patent, so what they actually do, they will take a patent for they will take the molecule patent. Then after that, after maybe either after 20 or 15 to 16 years, they will file another patent for the same molecule but a different form. So like there is crystalline forms and amorphous form. Both are the same. The molecule is the same, but they are actually claiming two different patents for the same molecule. So that is actually there is a situation called evergreening of patents. So that the patent terms never ends. They actually increasing the life of the patent, life of the molecule. Okay. So that is why we have introduced this particular section, that is mere discovery of a new form of a non-substance. So you are actually discovering a new form of a non-substance. Just imagine that there is a drug called sorafenib, and you are actually making a tosylate salt of this sorafenib. It's a salt form. Okay, you can say that. Just imagine this sodium acetate is a what? Sodium salt of acetic acid. Okay, both are the same. Agree? So both are the same. So if you are claiming like that, first you have claimed acetic acid. Now we are claiming sodium acetate. Okay. So that is not possible according to section 3D. It's a new form of a non-substance. Non-substance is acetic acid, and you are coming in with this particular new form, that is the sodium acetate. It is patentable if you are proving that this sodium acetate has got some therapeutic efficacy. You have to prove the therapeutic efficacy of that particular molecule. Then it is patentable. And mere discovery of any new property or new use of a non-substance. So there is a molecule which is actually intended for cancer. Now we are coming in with a new use of the same substance. Okay, you can say that aspirin. Initially, aspirin as which we usually prescribe for pain, right? Now we use it for what purpose? It is actually an anticoagulant. So we used to prescribe it to this cardiac patients, eccospirin. So this is actually a new property, right? New use of a non-substance. Non-substance is aspirin, and you are using it for a new indication. These things are not patentable in India, but in US it is patentable. There is new use patents are allowed. Okay, and so when you apply this to this biotech inventions, so how we can interpret? So invention related to three-dimensional crystal structure of polypeptides attracts the provisions of 3D. So you are claiming a polypeptide, and you are claiming with the new crystal structure of this polypeptide. Initially it was amorphous form. You are now claiming a what? Crystalline form. Okay. The crystal of peptide consisting of sequoid number A, wherein said crystal comprises an asymmetric unit. Said asymmetric unit comprises 4 molecules of said peptides per Zn2 plus, and further wherein crystal belongs to space group X, Y, Z. So these things are there. So the prior art was what? Amorphous form of the same sequoid. So the applicant failed to demonstrate any significant improvement in the property with regard to the therapeutic efficacy of the non-amorphous peptide. Okay. So while examining this invention, this inventor or the applicant can actually failed to prove the efficacy, the therapeutic efficacy. So whenever you are claiming a new form of a non-substance, you need to prove the therapeutic efficacy. Okay. So there is a non-drug, or actually this section deals with, actually this particular section is intended in the use of pharmaceutical field. So if there is a drug which is in the amorphous form, and now we are coming in with the crystalline form, and you have to compare the activity of the both the forms, and you have to prove that this crystalline forms has got some extra effect, that is a therapeutic efficacy. You have to prove the therapeutic efficacy. That is actually the Indian Patent Act mandates. The act, there is no mention of therapeutic efficacy, but in judgment they are saying that you have to prove therapeutic efficacy. Okay, that is section 3D. Then section 3E. So if you are mere admixture, just you are mixing two things together. Okay, just mixing two things together, and it's actually will not give any synergetic effect. Just imagine the situation, the synergism, right? So if you are 1 plus 1 is equal to 2, right? If you are 1 plus 1 is equal to 3, that is called synergism. So there should be some added effect. If you are combining two molecules or two ingredients, you are mixing two ingredients, and you are getting a result which is more than the effect of these two things when used individually. So that is called synergism, right? So if you are able to prove the synergism, it can be patentable. Otherwise, it is not patentable. Mere admixture resulting only in aggregation of properties. So it's just aggregating the properties of two things. Just imagine, the fixed dose combination drugs. So what is an example? What is Combiflam? Combiflam is a drug where it is actually, I think, paracetamol and ibuprofen, right? So paracetamol is having an effect, and ibuprofen is also having an effect, and you are mixing together, and they will act separately. Okay, but you can consume it as a single tablet. So that is the advantage there. You can consume it as a single tablet. Without taking two tablets at separate times, you can take a single tablet together. That's only the advantage. But they are acting separately. They are actually showing their effects separately. There is no enhancement of the effects. So such inventions are not patentable in India. So fixed dose combinations are not patentable in India. Okay. So here they have given an example, that is composition of innovative combination of dormant spore of naturally occurring Bacillomyces lilanis, I don't know. So, and Arthrobotrys fungus with enzymes, fats, and growth promoting molecules to control plant parasite nematodes. They're actually, mixing two combination of, one is a spore, one is a fungus, right? And it is found that the claim is directed to a composition of two non-fungal species, and the said two species used in alleged inventions are not for their nematode biocontrol activity. So these two are actually known for biocontrol activity. So they are actually biocontrol agents. Okay. The specification is silent on the advantage of the combinative effect of these two fungal species over the sum of their individual effects. So that is That's why it's not patentable. Okay. So these two drugs, actually, these two fungus species, they're actually, they're biocontrol agents. They're actually mixed these two things together, and they have actually claimed for a patent, and they haven't shown any additive effect. They're showing that these two fungal species they're mixed together and they're getting some effect. That's it. But they have to show the additive effect. That is, some synergetic data should be provided. Then only it can be patentable. Okay. Then the section 3H, that is the method of agriculture and horticulture, is not patentable under Indian Patent Act. So what is the conventional methods performed on actual open field should be considered as method of agriculture or horticulture. That is a method of growing leguminous plants as intercropping for improving fertility of soil by augmenting nitrogen content of the soil. So you know this thing, whenever you plant some coconut or something, or banana or something like that, you will plant leguminous plants like this pea plants. So as intercropping for improving the fertility of soil, so it will increase this fertility of the soil because it having this nitrogen consuming bacteria. I forgot that this actually. So there are some nodules in this root nodules. There are some bacteria. So it is actually increase the soil fertility. Okay. So this is a method of agriculture. It is already known. This kind of things are not patentable. Okay. Then method of treatment. So method of treatment is actually prohibited from patentability. So what will happen if you allow this thing? So everything will be very costly if you are giving patent to every fields of technologies like diagnostic things and everything. So even now it's also not affordable, right? If you are giving patents also, then what will happen? So imagine. So any process for the medicinal, surgical, curative, prophylactic or diagnostic methods are not patentable. So what is medicinal and curative and prophylactic methods? So medicinal method means, if you are claiming a method of administration of a tablet or administration of an injection, it's a medicinal method, or surgical methods, or the incision method, or curative method. Curative method means the removal of plaque from your teeth. And prophylactic, that means a prevention method, that is the vaccinations. That methods are not patentable in India. Okay. A method of monitoring drug response in a patient suffering from cancer treated with a combination of gemcitabine and P1446A, comprising dictation of gene signature with at least 2 drug response markers, wherein the said drug response markers are selected from the group consisting of these things. So just imagine this situation. So here they are actually claiming a method of monitoring drug response in a patient. So it's a method of treatment only. So these things are not patentable. Or method of injecting a medicine, or method of consuming a tablet. You are saying that you are claiming a method in which you are claiming that you have to take this tablet 3 times a day before food. This is a method of treatment. That kind of things are not patentable. Okay. So in biotechnology also that kind of things are not patentable. And section 3J. So what is actually 3J? Plants and animals in whole or any part thereof other than microorganisms, but including seeds, varieties and species, and essentially biological processes are not patentable. Okay. So from this reading, so you can say that they've actually excluded microorganisms, right? But including seeds, varieties, species, and essentially biological process. So you cannot patent plants, you cannot patent animals in whole, or you cannot patent any part of plant, any part of animals, other than microorganisms, but including so you cannot patent seeds, you cannot patent varieties, that the plant varieties or something like that, and you cannot patent species and essentially biological processes, but you can patent microorganisms, right? So that's what you can get from this particular section. But section 3 actually implies that what? Microorganisms are excluded from patentability. So now we can say that only the modified microorganisms are patentable. Okay. So they are in section 3C, they are saying that living thing occurring in nature are not patentable. So if a microorganisms are, we are actually modifying a microorganism. There is a microorganism and I am modifying a microorganism with a gene editing technology, a microorganism, not an animal, okay, just a microorganisms. So in that case, it is patentable. Okay. So claims related to essential biological process of growing plants, germination of seeds, development stages of plant and animals are not patentable. Okay, all these things are excluded from patentability. Clear? So then what is patentable? So we have actually discussed the non-patentable thing. So then what is patentable? So you can patent cDNA. Okay? And rDNA. And proteins, polypeptides, peptides. But you have to satisfy all these conditions. Okay? So if it is we have discussed, if it is a peptide sequence, you have to prove that it is novel, it is inventive, and it is having an industrial application, and it does not come under the sections, the excluded subject matter. Then it is patentable. And nucleotide, protein sequences, promoters, vectors, and living entities of artificial origin such as microorganisms, vaccines. Method of vaccination is not patentable, but vaccines are patentable. Okay, epitope sequence of antibodies, artificially produced cell lines, and process used for making the protein. So all these things are patentable. So can you say what all things are patentable here? A human baby, is it patentable? No, right? Because we have the morality issues, and a genetically modified human is also not patentable in India, and the animals also are not patentable in India. But we can patent this gene sequences and bacteria, which is genetically modified microorganisms. We can patent it. Clear? Any questions? So now I'll move on to I'm coming from this India University Center for IPR Studies. I would like to introduce the center to you. Actually, we are actually a state government-owned institute, which is situated in QSAT. So we have a direct funding from state government, and we have a governing council, and the chief minister is our governing council chairman. And we have some programs, education programs in our center. That is the PhD program, multidisciplinary PhD program is there. And we have 5-year integrated dual degree PhD program. That is LLM IP PhD and IPA PhD. And IP PhD is for non-law students, and IPA PhD is for law students. And we have a 2-year LLM course with a 15 student intake. And we have some facilities, we have some special facilities at our center, because it is actually a center for IPR studies. It's like only specialized for IPR studies. And we actually offers free advisory service for IP related matters. So till now we have catered around 200 plus individuals for this IP advisory service. And we have a facility for patent filing. We have a funding for patent filing for all the universities across Kerala. And till the grant we will fund them for all the patent related activities. And for the local individuals and local inventors and startups, so we have a consultancy which is at a very nominal cost. And we have a geographical GI working group. So there what we have actually do is that we used to facilitate the marketing and management of GI registered goods of Kerala. So till now we have did this for 2 products, that is Chendamangalam handloom and Pokali rice. And we have this assistance for filing of farmers variety in India, registration of farmers variety in India. Till now we have did 4 applications. And also we have a complete application of invention from initially we have only filing provisional application. Now we have started complete filing also. And this is the schedule of fee of our consultancy. So it's actually for novelty search, we are charging 5,000 for the individuals, and for the industries and universities, we are charging 7,500. And this is the fee structure. And this is the patent filing fee and patent drafting and filing fee. And these are the patents we have actually filed through our center. So for universities, we have filed around 37 applications, and which is out of which 1 is in US. And for QSAT, it is 27 applications. For Kerala University, it is 1 application. Kannur University, 3. Kerala Agriculture University, 3 applications. Calicut University, 2 applications. And KVASU, only 1. And the consultancy, we have actually catered for local inventors. We have filed 17 applications. And we have done novelty assessment for 15 applications. And out of this patents, 11 are granted and 1 is in US. Okay. So that's about it. So any questions?
Dear participants, the forum is now open for discussions. We might take a few questions. Dr. Rasha is ready to answer to any of your
I'm free
queries.
until 9. No issues. You can ask many questions.
Thank you, ma'am. Thank you.
If results
Ma'am, we do have a question here.
Yeah. If results of 2 components in particular combination showing enhanced result than their individual effect on a plant or animal, is it patentable or not? Yes, you have to prove that, if 2 combinations, that means if 2 components acting separately, so there is an effect, right? For if just say these 2 components are paracetamol and ibuprofen. So these 2 have 2 separate effects, and you are comparing it to a particular combination. Just say that 50%, 30% of paracetamol and 70% of ibuprofen, and you have to show that. So when you take a paracetamol tablet in 30% and 70% ibuprofen, there is an effect, right? So you have to show that this particular combination has got some enhanced effect when you take it separately. Then it is patentable, no issues. Did I answer your question? Dr. Anuradha ma'am? Okay.
We have Ms. Tisha Liza Tommy and Dr. Anasuya raising their hands for another question. You may please ask your question, ma'am.
Good evening, ma'am.
Yeah.
First of all, let me tell you, your presentation was like, it was really informative. Relentlessly, you were giving information to us. Thank you, ma'am.
Thank you, ma'am.
Ma'am, I have a doubt. Like in the first slides, you were telling about the monoclonal antibodies
Yes.
for cancer therapy. So, still, it's highly costly for, like, normal patients can't afford these monoclonal antibody therapies, right, ma'am?
Yes.
And if it is going to be patentable, whether this cost will get rise up or can be
Definitely.
definitely. So, what is the real purpose, ma'am? Like, we are doing something for the mankind, no? So after patenting something, how these medicine can be affordable to these normal people?
Yeah. It's a very good question, ma'am. Actually, this monoclonal antibodies are now actually gaining a lot of popularity these days. So because all these biologics. So why this cancer medicine has got the side effect? Because all these are chemical molecules. But when you go to this biologics are actually, derived from the living system itself. So they are not having any side effects. Okay. So that is why this biologics are gaining a lot of attention these days, because it's having less side effects. But the problem is why it is very costly, because the, the drug companies actually, what they actually do is, the development of a biologic is very difficult. In the case of normal drugs, what we actually do is, the drug development process, we actually do develop a drug, and we used to study the in vitro effects, like in the cell lines. Then we study in animals, in vivo studies. Then we conduct the clinical studies. So all these stages are there, right? And we can patent it also. If you want patent also, you can file a patent. And when a generic drug comes into the market, what they actually do is, they only have to show that this particular drug is chemically equivalent and biologically equivalent. That's it. They don't need to repeat all the clinical trial studies. Okay, that is with regard to the chemical molecules. But when it comes to biologics, what's the actual problem is, for the innovative drug, that is the first time if I'm developing a new biologic drug, I have to show all these things, that the in vitro studies, in vivo studies, clinical studies, everything. And when a company is wanted to introduce a copy of it, like a generic version of it, they have to also undergo all these things, because there is no this biologics and the generic version of the biology, that is the biosimilar, are not the same. They are similar, but they are not the same. So again, you have to do all these studies, this clinical studies and all these things. So that is why it's actually really costly. Even if it is not patented, it is really costly. But why we are giving patent to these kind of things? Because we are actually agreed to this TRIPS. India is actually agreed as a signatory to TRIPS. So we are actually obliged to to obey this rules of this TRIPS. So that is why we are included these things in patentability. But we have some provisions, when these drug prices are high, government can intervene. So there is, in one such situation, it happened once in India in 2011. So what happened was, there is a drug company called Bayer, where actually introduced a drug, that is sorafenib tosylate, and it is actually a 1 month treatment cost was around 2,80,000. Okay, and they have got a patent in India also, and they are not working the invention in India. They are just importing the drug from other countries. They have the patent in other countries also. They have a manufacturing unit in US also. They are exporting this drug from other countries, and they are selling it in India. So what happened was, there is a provision in our act, that is compulsory licensing. So if such a situation is there, any generic company can approach the government for a compulsory license from this drug company. So there is a company called Natco Pharma, based out of Hyderabad. They've actually said that they can offer this drug for 8,800 rupees against 2,80,000. So just compare the cost. So they have actually offered this price, and with a lot of negotiations happen, and finally they have got this compulsory license. So even now, just imagine the cost. So 8,800 for 1 month treatment. So how many people can afford that particular amount? This happened in 2011. So in 2011, 8,800 it's a much bigger amount, right? So how many people can afford that particular amount of drug price? So it happens, because that's why we have introduced these kind of mechanisms in our act itself. That is the government use provisions, the compulsory license provisions. But unfortunately, only once it happened. After that, this never happened. So it's a high level policy level things. So they are actually manipulating all these things. They won't give any compulsory license after they have actually decided that, but they have an agreement with these multinational companies. So that is actually happening these days. But we have to oblige to this TRIPS agreements. That's why we are giving patents to these kind of technologies. Is that clear, ma'am?
Yes, ma'am. Yes. Thank you, ma'am.
Yeah.
Ma'am, we have another query from Ma'am Mary Jyoti, madam, who says in case of COVID vaccine also, this compulsory licensing did not happen, even with the interference of the Supreme Court.
Yes, yes, it didn't happen because when I'm not saying against this government, but it happens. There is a news itself. You can search it in the Google. So they're saying that unofficially they were given a promise to US when Trump is the president. They actually Modi actually given a promise that we don't give any compulsory license from then onwards. There is a promise is there.
Then again,
There are a lot of drugs. Even now, it is very highly priced. This Herceptin itself, it is actually initially it was 1,13,000. Now it's only 59,000 or something. So that is also a huge amount, right? 14 such doses means how much is the money? That's the issue. It's still there.
yeah. And there was this query from Mary, ma'am. She wanted to know, so if can an Indian scientist apply for patency in other countries which give them permit?
Yeah, that I have already answered because you can
I
do
repeat.
that. Yeah. You can do that if you want to
All right.
patent these things in other countries, you can file in the different countries. There are different filing mechanisms. If you want to file a patent in different countries, say like in foreign countries, like America or in Japan or China, and you're not willing to file in India, that is also possible. But you have to take a permission from Indian Patent Office under Form 25. So you have to take a permission from the Indian Patent Office, then you can file a foreign country. Otherwise, the other provision is for foreign filing means you have to file in India, then within 12 months you have to file a foreign application. There are two systems, that is convention application and PCT application. These two are two foreign filing mechanism. So there is one more query. In many countries, there is an increase in smuggling of essential drugs because of this TRIPS agreement and American pharmaceutical industry. Yeah, it's happened because, we actually have a list of essential drugs. WHO already published this list of essential drugs. Based on that, we have actually prepared a list of essential drugs. Now in the new list, there are 4 patented medicine. So that is the situation. Initially there is a rule. There is not a rule. Actually, they mandated that this patented drug should not be included in essential list. And now there is 4 patented drugs, the new list. So that is why this smuggling is happening. You can watch a documentary in YouTube, and you will see the real face of pharmaceutical industry that is dying for drugs. And literally, you will cry after seeing that particular documentary. It's a real incident, not a film or something like that. It is real incident. Literally, you will cry after watching that particular documentary. It's dying for drug. You can watch it. So it will show the real face of pharmaceutical industries. And in that documentary itself, they are showing that they are smuggling drugs. The real life people actually smuggling drugs only because to save their people. That's it. It's a real situation happening here in African countries and all. They don't have this manufacturing capacities, so they used to depend other foreign companies. That's the issue.
Okay, thank you so much, ma'am. Any more queries? Any more questions, please?
Thank you, ma'am. There is Shamina, ma'am. You have any query?
Yeah, ma'am. Good evening, ma'am. I have query. Like you were saying about patenting in the other countries. So
Yes.
now let me ask, like, if it is a Indo-China, Indo-America patent, it's together. It's an inter-collaborative patent. So what about that, ma'am?
So what do you mean by this collaborative patent?
That is, people from China and people from India are merging together to file on a single patent.
Okay.
In
So
that
they wanted to file a patent.
Yeah, in that case, who will be is it like any importance to any country will be the majority, or what could be?
No, ma'am, it's not like that. If say for example, I am in India I'm collaborating with some US institute. We have a collaboration with the US Emory University. So if I'm collaborating with that university, and if you want to file an application together, so there you have to decide who is the applicant. Okay, if I am in QSAT, I am filing a patent from QSAT, and I am having a collaborator in Emory University and US. So if I wanted to file a patent in QSAT, QSAT is the applicant. Just imagine that situation. QSAT is the applicant. I am the inventor there, and this guy from this Emory University is also an inventor.
Okay.
Okay, so we need to get an NOC from this particular Emory University. Whether they have to stay that we don't have any objection in filing patent in the name of QSAT. Or either way, like if Emory University is filing patent there in US, and we have to say that we don't have any objection. Okay.
Okay.
Otherwise, you can apply as co-applicants. That's another option.
Okay.
It's actually not the country's decision. It's actually with the applicant's decision.
Okay. Okay. Thank you.
Okay.
Any other queries, or shall we proceed with the vote of thanks? Anybody else?
Somebody has raised a hand.
Yes.
Yes, ma'am.
Senthil Kumar, sir.
Yes, sir. Proceed,
Yes.
sir.
Madam, good evening. Good evening, madam. Very nice presentation from your side.
Thank
I
you.
have few questions, ma'am.
Yeah, tell me.
Number one, there is why Indian patents are get delayed when you once you file the patent. It normally it's get to take a lot of time, even the examination, publications, and all. When compared to international office, why this time-taking process happen in India? Number one. Question number two. Many of the computer-based analysis get patented from international level, but it is not in India. Number two. Number three.
So I didn't get you. So computer-based?
Your institute is offering two computer-based
Sorry, sir.
in silico analysis, especially the biotechnology.
Okay.
Can you hear, ma'am?
Yeah.
Yeah.
What?
The computer-based in silico
second
analysis,
question.
especially in biotechnology. That is, many of the in silico, that is a computer-based analysis, get international patents.
Yeah.
But how they allow these in silico analysis, the computer-based analysis by international office?
Okay.
Number two. Number three, your institute offering 2 years program on the IPR program. Is it sufficient to be act as a patent lawyer, or after the program should they qualify themselves to undergo any examination conducted by patent government of India? These are all the few doubts, madam. It's a common problem, because you are the right persons to answer all these questions, ma'am. Thank you.
Yeah. Thank you, sir. So, with respect to your first question, that is the delaying in grant of the patents. So we have a provision that the expedited examination and early publication request. So usually in Indian Patent Office, what is the procedure means? When you file a patent application, and in normal way, actually, if you file a patent application, in 18 months it will publish, and after that they will examine the application. Okay, provided you have filed a request for examination. And what is the delay? Because patent office have not enough capacity of this examiners. So now they have actually called for this exam. For 550 vacancies are there now. They're actually in short of examiners. That is why the delay. So they have actually capacity in some areas. In some areas they don't have capacities. Some engineering inventions, they don't have capacities. So we have filed a patent, and it is actually we have done everything. Like we have answered the queries, and we have responded to FER. We are attended the hearing. Everything happened, but after the hearing, it's actually elapsed 2 years. Till now, it is not granted. So it is the behavior of the patent office. I don't know why they are behaving like that. It's only because of the lack of capacity, lack of the examiners. And for this actually there is the provision of expedited examination. If you are from a government institute, or if you are having a women inventor, or if you are a natural person like natural inventor, you can avail this particular provision, that is the expedited examination request. So normal way it is published in 18 months. If you are filing it in expedited way, it will publish the very next Friday after your filing, and after that it will go for the examination, and they will conduct all the other process. Okay. And we have done this for many applications till now, and one of the patent which is granted is within 9 months. Okay, so usually it grant within 2 to 3 years, but it is granted within 9 months. The 2 to 3 years is the normal time, and 9 months is actually happened because we have filed an expedited request. So, but even in expedited request also it got delayed. So last application we have filed, which is after 1 year also it is not granted. We have filed an expedited request also, but it's not granted till now. So it's because of the capacity of the patent office. Okay, so that is your first question. Second question, the computer-based in silico analysis. So I'm not clear about your question, because I understand that this in silico based sequences are patenting, right, sir?
Yes, ma'am. Yes.
Yeah.
Yes.
Yeah. So provided if you are what, there are a lot of programs in silico models are there. You can design a lot of sequences. So based on the sequences, you have to prove that. So in some of the sequence, you have to prove that there are some function. Then only it is patentable. Without giving this function, but simply designing these sequences and filing a patent, it's not possible. That's what my understanding. You have to prove a function for this particular sequence, which is actually designed in silico. That's what my understanding, sir. And based on your third question, that is a 2-year LLM course. It's actually for LLB graduates, not for any other graduates. It's actually for LLB graduates. They can undergo this course, and they can become an IP lawyer. Okay, not a patent agent. They can become an IP lawyer without undergoing any other course. They can become an IP lawyer. But for to become a patent agent, they cannot write this exam for patent agent if they don't have a science degree. So for writing a patent agent exam, you should have a science degree. That is a basic qualification. If you have a science degree, you can undergo this particular patent agent exam. For patent lawyer, there is no exams. Is that clear, sir?
Okay. Thank you very much, ma'am. Thank you. Thank you, ma'am. Thank you. Thank you very much.
Thank you, sir.
So, are there any more queries? Any further doubts? If not, this is indeed a pleasure today to call upon Dr. S. Shamina, Associate Professor and Head Department of Biochemistry, RVS College of Arts and Science, Salur, Coimbatore, for the word of thanks. Ma'am, Dr. Shamina, ma'am, over to you.
Thank you, ma'am. Am I audible?
Yes, very.
Good evening to all of you, respected ladies and gentlemen. As we come close of the end of the third day session of our 7-day FDP on intellectual property rights, on behalf of our organizing team, it's my privilege to propose a vote of thanks on this FDP session. My heart fills with love of gratitude and respect for our respected and distinguished guest speaker, Dr. Asha Ma'am, Research Officer, Inter-University Center of IPR Studies, Kushat, for sparing her valuable time for us and enlightening us with her commendable talk on the topic, patentability of biotechnological inventions. Ma'am, your session was so clear and it has empowered us with the knowledge of biotechnological inventions. The way you started with the scratch as to start with what is an invention, what is a patent, what are biotechnological inventions, its novelty, its criterias, industrial applicability, unity of inventions, and the claims. Overall, it was a valuable insights which enriched us so much knowledge regarding the session. Thank you once again, ma'am, for clearing all our doubts and introducing the center of yours, which is the Inter-University Center for IPR, which gave us knowledge what can be done in near future. I also thank all the participants for their active presentation in the FDP. Thank you for the entire organizing team for organizing the FDP and the DPT STAR scheme, which paves way for the teaching fraternity to enter into the research aspects. Thank you once again. Meet you all once again tomorrow at the 4th day. Stay tuned.
With that, we come to the end of the third day sessions. Thank you so much, all the participants, the guests, and other organizing committee members. Have a wonderful, restful night before we meet again for day 4's session tomorrow at 7 p.m. sharp. Good night, everybody.
Thank you, ma'am. Thank you so much. Thank you all.
Thank you, ma'am.